Alright guys, so hopefully you just took a quiz from the start of cellular respiration all the way to the citric acid cycle. And we are going to finish aerobic respiration today with the electron transfer chain and chemiosmosis. So you already have this diagram. It's on the back of your Krebs cycle diagram, so have that out. And while you have that out, I'm going to go ahead and start labeling some stuff for you. Alright, so this should look familiar. You've seen this before in photosynthesis, and in cellular respiration there's a very similar electron transfer chain. But one of the first things I want you to notice that up here, yes, this is the mitochondria, and this is happening in the mitochondria. Notice that the dark orange color corresponds with the top of the graphic, and the light yellow color corresponds to the bottom of the graphic. This light yellow color is called the matrix, and this is the mitochondrial matrix. The dark orange color right here is the intermembrane space. This is super important because we're going to talk about a concentration gradient in just a second. So know the difference between the matrix and the intermembrane space. Now from the mitochondria worksheet, you should remember the folds inside of the mitochondria, so all of these folds you see have a name. That is shown right here, also called the inner mitochondrial membrane, but you should know it as cristae. Those are the folds of the mitochondria, and that is where you will see this phospholipid bilayer and these proteins. So let's go ahead and label the proteins that make up our electron transfer chain. So all of these purple structures, 1, 2, 3, 4, 5. These 5 proteins are part of your electron transfer chain. So I'm going to go ahead and put an arrow right here for you guys. This is your ETC. Now, you already know from the Krebs cycle you have some electron carriers coming in. The whole purpose of the Krebs cycle was to create these electron carriers so they can fund the electron transfer chain. Okay? So from this graphic it should look familiar. You already know what the very first electron carrier or electron food is coming in is NADH. He's going to leave as NAD plus, because what do you think we do with the hydrogen? We steal its electron and send the positive hydrogen to the matrix. And this is happening over and over again. So the matrix is loaded with hydrogen ions without their electrons. But we also want to show that these electrons are traveling in this yellow arrow. You do that by putting little e's with a negative sign next to it. And these are traveling through this yellow arrow. This yellow arrow, from the beginning to the end, is the electron transfer chain. So you have a bunch of hydrogen ions coming inside, right? Because of NADH bringing them in. The problem is that you have created a concentration gradient. So now these hydrogen ions naturally want to go into the inner membrane space because there's a concentration gradient. And on these proteins, you see these pink arrows. That is the direction they take to get out of the matrix and into the inner membrane space. So go ahead and on these arrows, you can start drawing hydrogen ions, and these gray circles are hydrogens leaving the matrix and going to the inner membrane space. So you go ahead and draw some hydrogen ions in the inner membrane space as well. Now, this is happening at such a fast rate that these hydrogen ions are going to want to come back in. And just based on this diagram, I'm sure you can already tell how he's going to come back in. You've seen this before. We dealt with this enzyme before. He looked just like this, kind of looks like a doorknob. His name is ATP synthase. You know he's an enzyme because he ends with ACE. I'm going to bold this. This is ATP synthase. Okay? And you guessed it. He's going to take in all of those hydrogen ions coming back in. So this is a hydrogen ion. They come back in. And as they're coming back in and they're oxidizing, they're creating and releasing energy to phosphorylize adenosine diphosphate with an inorganic phosphate group to create ATP. So I'm going to do that in a different color for you guys so you can see it. All right, so right here we have ADP plus inorganic phosphate making ATP. I'm going to bold that. Now, you should remember that the electron transport chain is where we get majority of our ATP from. And this range is anywhere between 32 to 38 ATP molecules. Now, I always get asked, why is there a range? And one of these range, and there's not a spot to write this down, is because of our second electron carrier, which is FADH. FADH is a lot slower than NADH. If you remember from the Krebs cycle, you have 6 NADH molecules and just 2 FADH molecules. So it takes FADH a little bit more time, but also a little bit more energy to get to the electron transport chain. So if he gets here on time, he enters this protein here, and then of course leaves at FAD plus and goes back to the Krebs cycle. However, if he shows up later and needs to enter this protein, that's how you get less ATP. So the earlier he comes and enters the electron transport chain, the more ATP you get. The later he shows up, the less ATP you get. And this directly correlates case-by-case basis on people, right? What is our ability to inhale oxygen, exhale CO2? What is our rate of respiring? How much energy do we use? How many calories do we burn? And that will determine on a biological level when the FADH will enter the electron transport chain. Now, there's another thing that's not accounted for on here, is the oxygen. How are we inhaling the oxygen? Where is the water? Where are the products? So, if you'll notice, there is a black arrow coming out from the end of the electron transport chain. Does everyone see that? Right here, you're going to have oxygen come in. And oxygen is the very last acceptor of this electron. And now remember, NADH continues to come. So that's why you have this backup of electrons here. And oxygen wants that electron. However, when he comes through, when we inhale, this is how we breathe, folks. So when we're inhaling that oxygen and we're about to exhale, it's going to bond with these 2 protons right here. So how do we get water? We exhale it as water vapor. Is because as the oxygen comes in, he picks up that very last electron. These 2 hydrogen ions bond with it. And this is why water is in the products of the cellular respiration equation. We exhale it. Now this is really interesting because this is happening with oxygen. This, on a biological level, is how poisoning works too. So if cyanide, if you remember this from the Amoeba Sisters video, if cyanide was to enter the bloodstream, wherever this O2 originally comes from to go to this electron transport chain, cyanide is so much more powerful than oxygen that when it comes in, it grabs not only that electron, but that hydrogen and pushes these oxygen molecules out. So if you ever watch any of those crime videos or any of those detective videos and you see dead bodies that have been through cyanide poisoning, You'll notice that their nails are blue, their lips are blue. That's because the oxygen is literally pumped out of the bloodstream. And when that oxygen is pumped out, cyanide is actually a blue colored poison, and that's what takes over. So how poisoning works, it disturbs the electron transport chain of the cellular respiration. So it actually disturbs the breathing process of these gases being inhaled like oxygen and CO2 and H2O being exhaled as water vapor. So that's the electron transport chain. Over here I have the graphic filled out a little bit more neatly. The only thing you will notice that it doesn't really do a good job of showing the surplus of hydrogen ions, right? This is super important because the surplus is what winds this ATP synthase and creates that energy like a hydrothermal dam, right? This is where that heat is created from to phosphorylize that ADP. So just if you are going to study from this graphic, please draw extra hydrogen ions to show that movement, okay? Remember that this is where H2O is also created, because as the O2 enters our bloodstream, it picks up these two hydrogen ions and is released as water vapor. All right, so moving on. Right here. Now let's take an overview of just aerobic respiration. Remember that all processes of cellular respiration begin with glycolysis. It doesn't matter if you're going to do aerobic respiration or anaerobic respiration. It has to begin with glycolysis, where the starting point is glucose. You convert that glucose into two pyruvate or pyruvic acid molecules. Those enter the mitochondria to make two acetyl-CoA molecules. Further enter the mitochondria into the citric acid or the Krebs cycle to provide enzyme carriers like NADH and FADH. And those then fund the electron transport chain to give us the most amount of ATPs. And hopefully, if we do this correctly, the max amount of ATPs we can get are 38. But let's say we can't do that. Let's say oxygen is not present. What do our cells do? This is where anaerobic respiration comes in. Now remember, our cells don't like anaerobic respiration because they think of ATP as currency. You do not make enough ATP in anaerobic respiration. You make the most of it in aerobic respiration. That's why they prefer to do aerobic respiration. Now, it's us who make the decisions to go through anaerobic respiration. Like if humans were to do that, like weightlifting would be one way, right? And that is when lactic acid is released into your bloodstream and you're actually tearing your muscle tissue, and that's why you feel sore for the next couple of days. So that's a choice we make, and our cells don't prefer that. Our cells are always going to want to maintain homeostasis, always going to want the most amount of ATP, which is again through aerobic respiration. Now, we aren't the only ones that do anaerobic respiration. It is done by yeast and bacteria. Even when we do anaerobic respiration, it's the bacteria that lives inside of us that does it for us, okay? We biologically cannot do that. It's our Streptococcus bacteria that will do it. Now, another thing you need to remember, because this is anaerobic respiration, meaning it's happening without oxygen, it's not happening in the mitochondria. It's happening in the cytoplasm. Okay? Again, this starts with glycolysis. So you should, or the sub should at this point, have passed out the anaerobic respiration diagrams. It's two-sided. Please make sure you're looking at the side that has just 2 molecules on it and not 3. I'll go ahead and show that to you right here. This is 1 molecule. This is 2. Both sides look very similar because fermentation is very similar like that. So please make sure you're on this side. And if the sub has not passed it out yet, make sure you get the papers from the sub. Now, the best thing about learning fermentation is that you already know most of it because 90% of the step is glycolysis. And remember, we've already covered glycolysis. You already know glycolysis. So on this graphic, you know 95% of what's here. The only thing that's different is the final product. But you know what? You already know the final product too, because it's called lactic acid fermentation. You're fermenting lactic acid. This is what we do. This is also what bacteria do to make yogurt. This is what bacteria do to make sour cream. This is the process that it's made in. So it starts off with glucose, just like in glycolysis. This arrow is representing the process of glycolysis. And if you recall from glycolysis, we get 2 ADPs that bond with 2 inorganic phosphates to give us 2 ATPs and also 2 pyruvate or pyruvic acid molecules. And these boxes you should be familiar with are our electron acceptors. So we have 2 NADHs in glycolysis, which lead us to 2 NAD+, because these 2 hydrogens are going to leave. Okay? Now, this is pretty simple. Take a look at this product and compare it to the pyruvic acid. Structurally, you'll notice only 2 things have changed, and I bet you can figure out where those changes came from. You'll notice that there are 2 hydrogens added to the pyruvic acid to alter its form. Those 2 hydrogens came from NADH. NADH, the electron carrier, donated its hydrogens to pyruvic acid. When that rearrangement happens, you create lactic acid. In chemistry, we call lactic acid lactate. In biology, we call it lactic acid. Everything is 2. We start off with 1 glucose, which gives us 2 ATP, 2 pyruvate molecules, 2 NADH, 2 NAD+, to give us 2 lactate molecules. So that is lactic acid fermentation. Now, the second type of fermentation you're going to learn is called alcoholic fermentation. Now, we do not do this, okay? But bread is made through alcoholic fermentation. Beer is made through alcoholic fermentation. Wine is made through alcoholic fermentation. And it's very similar to lactic acid fermentation, except you'll notice that there is an intermediate step. Okay, so let's go ahead and circle all the main characters in this diagram. So we have here, we have 1, 2, and finally 3. You should be able to fill this out. We just did it on the back side. Go ahead. As I fill it out, I want you to fill out what you remember. Again, this is just glycolysis. So here we have glucose. This is glycolysis, which gives us 2 ATPs. Those come from 2 ADP molecules, bonding and phosphorylizing with 2 inorganic phosphates. That gives us 2 pyruvate molecules. Now, what you don't know is this intermediate step. This intermediate step, take a look at what is happening to these atoms compared to these atoms over here. You'll notice something has left, okay? So over here, this no longer exists. You'll see that there's only 1 left. So it leaves as CO2. So the very first waste product is CO2, and specifically 2 CO2 molecules, okay? This is why there are bubbles in wine. This is why there are bubbles in beer. This is also why when you cut open a piece of bread, you see small holes. They actually come from the CO2 being released. So the biggest difference between alcoholic and lactic acid fermentation is the release of CO2. Now, the reason why you don't get drunk when you eat bread is because the heat is killing off that alcohol, but the bubbles stay. Whereas in beer and wine, those bubbles are still there, and that is because of the CO2 that's being released. The yeast will release CO2 as a waste product. Again, you have two NADH molecules here that will donate hydrogens, specifically 2 hydrogens, and leave as 2 NAD plus to go through this process again. Now again, compare this final product to this one here. This guy here has a name. You don't need to know his name, but I'm going to label him just for explanation's sake. This is going to be 2-acetylaldehyde. Acetylaldehyde will rearrange itself with the help of NADH in donating those 2 hydrogens right here and right here. You'll notice this double bond just breaks. And when this double bond breaks, it bonds with that positive hydrogen to give us 2 ethanol molecules. Ethanol is a type of alcohol, and it's the specific type of alcohol that will ferment into wine, beer, and bread. Okay, so that's alcoholic fermentation. Now, moving on, I want you to take a look at this graphic. Notice that today we only covered this fermentation and this fermentation. But there are so many other types of fermentations that we don't cover. Know that they exist. That's all you really need to know. But this is the most asked question I get is, why is rubbing alcohol a different type of alcohol as compared to beer and wine? Because if you look at it, yeast is what ferments wine and beer, whereas a bacteria ferments rubbing alcohol, acetone, nail polish remover. All of that stuff is fermented by a bacteria. Vinegar is fermented by a bacteria to make acetic acid. Acetic acid is the chemical name for vinegar. Notice even cheese has a specific bacteria that's fermenting it. That Swiss cheese, the holes exist because of CO2. You see it right there. Those bubbles here exist because of CO2. So all of these waste products actually have a physical difference on the final product. Notice none of these other products are bubbly because they don't release CO2. All right, so that wraps up cellular respiration. We've covered anaerobic respiration with lactic acid and alcohol fermentation, and aerobic respiration with everything from glycolysis to the link reaction to the Krebs cycle, all the way till chemiosmotic and electron transfer chain. Now, your quiz for this will be on Monday, the 25th. So make sure you're prepared from everything from the electron transfer chain to the end of anaerobic respiration.